This is the podcast for STEM Biology, Chapter 24. We're skipping ahead on viruses. There are two sections. First section is the most important. That is on viral structure and replication. Second section is on viral diseases. First, I want to show you a picture of what a virus can do. On the left, you have a healthy tobacco plant. Nice green, smooth leaves. And on the right, you can see there's patches of something that's over that leaf. That is caused by a virus. Viruses are specific. The tobacco mosaic virus, or abbreviated TMV, is a specific virus that affects tobacco plants. And as you can see the results of what that can do. Some history here. In the late 1800s, scientists didn't know what a virus was. There was no technology around for them to even look at them. Eventually, they found out that viruses aren't bacteria that's causing the diseases. They knew it was smaller than a bacteria cell, and so they began to study viruses. And what they wanted to know was if they were made of cells or not. So that is the big question. Are viruses made of cells? The scientist that found out, his name is Wendell San Lee, and what he did is he took that TMV plant and he broke it down to its smallest part. Okay? And so he was expecting to see a cell, the smallest unit of life, but instead he saw crystals. They look like that. And so he concluded that viruses cannot be alive because having a cell is one of the characteristics of being a living thing. So viruses are not alive. These are the characteristics of viruses. Viruses do not have cytoplasm. They do not have organelles like ER, Golgi body, etc. They cannot do metabolism. They can't do homeostasis, and all of these are properties of life. They don't have cells. They do have, however, DNA or RNA. They don't have both, but they do have a nucleic acid. And they cannot reproduce on their own. They cannot reproduce it unless they're in a host cell. They have to enter a living cell and use its machinery in order to reproduce. Sizes of viruses. Viruses are extremely small. They cannot be seen using a light microscope, so we will not be looking at viruses under our microscopes. They vary in size and shape. For example, the TMV, those ones right there are helical. They're kind of like rod-shaped. They call those helical viruses. Then you have an animal virus, like adenovirus, which is a virus that causes the common cold. That shape is called polyhedral. And then you have the bacteria, the viruses that affect bacteria, bacteriophages, and that shape is called icosahedron. Virus structure. There is a capsid. A capsid is a protein coat that surrounds the DNA or RNA. There is an envelope. Some viruses will have the second layer over the capsid. It is made of specific proteins and helps new viruses recognize their hosts. And bacteriophages will have a tail and tail fibers. Remember, bacteriophages are viruses that attack bacteria. So here is a diagram. Here is a bacteria. This is for the bacteriophage. You have your DNA or RNA inside the capsid. And the capsid could be polyhedral, icosahedron, etc. So this is a protein coat. Some of them will have an envelope around this as well, not bacteriophages usually. Underneath that capsid is a neck and a collar, and then they have a tail sheath, and then attached to the tail sheath are tail fibers, and then underneath the tail sheath is a base plate. Viruses can be classified based on these things: whether they have RNA or DNA, whether the RNA or DNA is single or double-stranded, whether the RNA or DNA is circular or linear. They also can be classified based on the shape of the capsid and whether or not they have an envelope around the capsid. Other things to know. Outside their host, viruses are lifeless. They have no control over their movements. Viruses can be spread through many things. Spread through the air, water, food, and bodily fluids. In order to replicate, viruses need to recognize their host cell before they can infect it. An enveloped virus needs to match the host cell's membrane receptors precisely. Otherwise, it can't get in. Okay, so how do viruses replicate? The DNA in some DNA viruses will enter a host cell and make it mRNA. The DNA in some DNA viruses insert into the host cell's chromosomes. If it's inserted into the host chromosomes, it's called a provirus. Some DNA viruses use the host cell enzymes to make new viral DNA and eventually new viruses. And that's the one we're going to look at in a minute. Replication in RNA viruses. Some RNA viruses enter the host, and then the RNA will serve as mRNA, which is then translated into new viral proteins immediately. Some RNA viruses, called retroviruses, contain a special enzyme called reverse transcriptase in addition to RNA. Reverse transcriptase uses RNA as a template to make DNA, which is then inserted into the host cell's genome. This is a very serious type of virus that does that. Reverse transcriptase reverses the normal process of transcription. We will be learning specifically transcription in the second try. HIV, for example, is a retrovirus, and because it can take RNA and make DNA from it, that's what makes it such a serious virus. Okay, how does a bacteriophage replicate? This is what we're going to learn. Remember, bacteriophages are viruses that attack bacteria, and they have complex capsids. There are two different ways that bacteriophages can replicate. One is the lytic cycle, two is the lysogenic cycle. We're going to look at lytic cycle first. For the lytic cycle, remember, lytic means lyse or break. Just like hydrolysis was breaking the polymers into the monomers, lysis means break. Lytic cycle means break. So what's breaking? That means that the host cell is going to break. The host cell is going to rupture after replication is completed. Viruses that do this cycle have a special name called virulent. There are 5 steps to the lytic cycle. Step number 1. The bacteriophage is going to attach its tail fibers to specific receptor molecules on the cell surface of a bacterial cell. So basically, now step number 1 is attachment. The DNA will then be injected into the host cell. The ends of the viral DNA attach to each other, forming a circle. It is separate from the host cell's DNA. Number 3, the viral DNA takes control of protein synthesis, so proteins are being made, and the viral genome, or DNA, is being copied. So basically copying DNA and making proteins. Enzymes and ribosomes are used from the host cell. New viral pieces are then made and put together. Number 5, lysozyme enzymes digest the cell wall, and up to 200 new viruses then are released. Here is a rough picture of the lytic cycle. So here is part A. Here is your drawn picture of a bacteriophage attached to the bacterial cell, and it's inserting its DNA inside. The DNA forms a circle around. Using the bacterial cell's machinery, ribosomes and enzymes, we get new viral protein capsids are being made, the DNA is being copied, and then eventually enzymes, lysozymes specifically, are going to break this wall down, and we get 200 viruses being released. Another way that bacteriophages replicate is called the lysogenic cycle. Basically, the same thing. There's a couple little differences. First difference is the host cell does not get killed in the end. It allows viruses to hide in the host cell for days and days and days, months, or even years. A virus that does this cycle is not called virulent, it is called temperate. There are also 5 steps to the lysogenic cycle. Number 1, the bacteriophage attaches to the host cell and injects its DNA inside. 2, the viral DNA attaches its ends to make a circle, so there's nothing new yet. This is where it's different. Number 3, the viral DNA then integrates and mixes in with the host DNA, and now it has a special name. It's called the prophage. When the bacteria replicates then its own DNA, it'll also be replicating the viral DNA. So now each bacterial cell will be infected with a prophage. The prophage will then exit the host cell, usually caused by radiation or chemicals. All right, so here's your lytic cycle. Again, bacteriophage attaches to the cell wall, DNA is inserted, The DNA forms a circle, viral pieces are made and replicated, and then the host cell breaks open, viruses are released. That's lytic. Lysogenic, the virus attaches to the cell wall, viral DNA comes in. The viral DNA will make a circle. But notice in the lysogenic cycle, the DNA from the virus attaches and connects to the DNA from the bacteria. And so when the bacteria is doing their cell division, the DNA from the virus is also getting replicated. And so eventually viral DNA is integrated. That's called the prophage, and then it just keeps going over and over and over again. And then sometime through radiation or some type of chemical reaction, the lysogenic cycle will end. All right, so where did viruses come from? Most scientists think, so this isn't for sure yet, that viruses originated from naked pieces of DNA or RNA that evolved from that. Viruses mutate quickly, such as the flu and HIV. It is difficult for the immune system to recognize and destroy them, which makes it hard to make good vaccines for a virus. A vaccine is a live virus that you get injected so that the body can make antibodies to kill it. So that's why you get the flu vaccine every year, because it mutates so much. You can't just get it one time and be done with it. Therefore, each year there is a new flu shot available. And that ends podcast chapter 24, part 1.